Welcome to this introduction to the MeRIP-seq learning module. This module was developed by the Bioinformatics Core at the University of Hawaii at Manoa within the Department of Quantitative Health Sciences. It is a cloud-based, hands-on resource designed to guide users through the analysis of RNA methylation, specifically M6A, using MeRIP-seq data. This workflow emphasizes reproducibility and accessibility, supporting both beginners and intermediate users in bioinformatics. Let's begin with RNA methylation. RNA methylation is an important layer of post-transcriptional gene regulation, also called epigenetic regulation. Among more than many known chemical modifications on RNA, N6-methyladenosine, or M6A, is the most abundant internal modification in eukaryotic messenger RNA. These modifications are not limited to mRNA. RNA methylation can also occur in tRNA, rRNA, snRNA, SNO RNA, microRNA, and even viral RNA, affecting a broad spectrum of RNA types across biological systems. Let's now focus on N6-methyladenosine, or M6A, the most common internal RNA modification in eukaryotic cells. It involves adding a methyl group to adenosine's 6th nitrogen. The M6A methyltransferase complex, or writers, installs M6A typically at GGACU motifs. Eraser proteins can remove it, enabling dynamic control. Reader proteins bind M6A, influencing RNA stability, translation, and splicing. Overall, M6A acts as a post-transcriptional switch, tuning transcript fate based on environmental and developmental cues. To study M6A modifications at a transcriptome-wide scale, one of the most widely used techniques is MeRIP-seq, also known as M6A-seq. MeRIP-seq combines methylated RNA immunoprecipitation with high-throughput sequencing. The process begins with the fragmentation of total RNA, followed by immunoprecipitation using an antibody specific for M6A modified RNA. The immunoprecipitated RNA, which is enriched for methylated fragments, is then sequenced in parallel with an input control sample. Mapping the reads to a reference genome allows us to identify regions of enrichment which correspond to M6A sites. This technique enables the identification of M6A modified regions, known as peaks, across the transcriptome, providing both localization and quantification of methylation marks. Although MeRIP-seq does not achieve single nucleotide resolution, it has become the foundational method for epitranscriptomic studies and continues to be widely used in both basic and translational research. Now let's talk about how to analyze the data generated from MeRIP-seq. MeRIP-seq data analysis typically follows a structured workflow designed to detect and interpret RNA methylation patterns across the transcriptome. The process begins with raw sequencing data in the FASTQ format. Early steps include quality control, alignment to a reference genome, and formatting to prepare for peak detection. Peak calling algorithms are then used to identify M6A enriched regions by comparing immunoprecipitated samples to corresponding input controls. This is followed by differential methylation analysis, which compares methylation levels across experimental conditions such as healthy versus disease states. Next, further downstream analysis links these methylation changes to biological functions, for example, through functional enrichment analyses such as gene ontology or KEGG pathway mapping. One of the unique features of MeRIP-seq data is that gene expression information can be extracted directly from the input control samples. Because these libraries are not enriched for M6A, they reflect the baseline transcriptome. This makes it possible to integrate methylation and expression data within the same dataset, enabling researchers to explore the relationship between M6A modification and transcriptional regulation. Despite its utility, MeRIP-seq presents several analytical challenges. These include variability in immunoprecipitation efficiency, RNA composition, and the need for careful normalization. Robust statistical models are required to distinguish true methylation signals from background noise. As far as we know, there is no widely adopted standardized analysis workflow, which can be a barrier for new users. To address this gap, our team has developed a reproducible, cloud-ready learning module that guides users through the complete MeRIP-seq pipeline. Structured across several modules, it provides an end-to-end framework from raw data to interpretable results. In this MeRIP-seq learning module, we begin with raw sequencing data in FASTQ format. Users follow a guided workflow to process these sequences, quantify RNA methylation levels, and identify differentially methylated regions and pathways, as well as perform other downstream analyses. All materials are provided as interactive Jupyter notebooks, which are available for download from the NIH-supported GitHub repository. Example datasets are included in cloud storage to support hands-on learning. To run the analysis workflows, users will need access to a cloud computing platform such as Amazon Web Services or Google Cloud Platform. The notebooks are optimized to run on AWS SageMaker and Google Cloud's Vertex AI. In this video, we'll demonstrate how to use this module in AWS SageMaker. This diagram provides a detailed view of the learning module's architecture, illustrating how the analysis flows across cloud-based components. We begin by downloading FASTQ files from a public dataset, trimming them to a smaller size for demonstration purposes, and storing them in an Amazon S3 bucket. Submodule 1 performs data pre-processing, which includes quality control, aligning reads to a reference genome, and performing peak calling to identify regions enriched with M6A, all using a Python-based Jupyter notebook. Gene expression analysis is also performed based on the input samples, treating them as RNA-seq data. The outputs from this step include BAM files for alignment and a gene count table in text format. In Submodule 2, the BAM files are then used to perform differential methylation analysis across experimental groups. Several algorithms implemented in R are introduced and compared in this step to highlight different statistical approaches for identifying M6A changes. The output is a bed file containing annotated M6A peak regions. Submodule 3 uses the bed and gene count files to perform downstream analysis based on the R environment, including functional enrichment, gene expression integration, and visualizations such as volcanoplots, metagene profiles, and coverage maps. Submodules 4 and 5 introduce workflow automation using Nextflow, built according to NF Core standards. These modules integrate the pre-processing and differential analysis steps from earlier submodules into a single, reproducible pipeline. Submodule 4 allows interactive execution, while Submodule 5 demonstrates how to deploy the pipeline using AWS Batch, which provides scalability and automation for larger datasets or recurring workflows. To begin using the MeRIP-seq learning module, visit the GitHub repository linked below and follow the setup instructions provided in the README file. Once your environment is set up, launch an AWS SageMaker notebook instance and download the module from GitHub. The downloaded learning module is organized as a structured series of Jupyter notebooks, each corresponding to a key step in the MeRIP-seq analysis pipeline. Users will see these files listed in the file browser on the left-hand side of the SageMaker interface. In addition to the README file, key components include Submodule 0, a PDF file that includes the biological background of RNA methylation and the MeRIP-seq technique. Submodules 1 through 5 are provided as a Python or R-based Jupyter notebook. Users can open these notebooks directly in the Jupyter interface to interact with the content. Next, let's take a look at how these notebooks are structured and how to use them effectively. Each Jupyter notebook in the module is designed to be self-contained and interactive. Inside the notebook, you'll find a combination of written instructions, executable code, and in some cases, previously generated outputs. Each notebook contains two main cell types, Markdown cells and code cells. Markdown cells include explanations, guidance, and contextual information. Read these carefully to follow the intended analysis flow. To run a code cell, click the Run button at the top of the interface, or press Shift and Enter. The output will appear directly beneath the code block. If an error occurs, the corresponding message will also be displayed in the output area. Some sub-modules include interactive components such as quizzes and flashcards to reinforce key concepts and improve long-term retention. We encourage you to explore the notebooks at your own pace, try running the code, and engage with the interactive content to get the most out of the learning experience. Thank you for watching this brief introduction to the MeRIP-seq learning module. We hope this module empowers you to explore RNA methylation analysis with confidence and curiosity. Enjoy learning!